Okay, we're going to talk about mitosis. Mitosis is important because it is the process through which our cells are going to allow us to grow, develop, and repair our bodies over time. So the two major purposes for mitosis are for growth and repair. Growth meaning that when you were a baby, you were made up of a certain number of cells, and when you're an adult, you have more cells. You've grown, you've gotten more cells in your body, makes you bigger. Your cells don't get bigger to make you bigger. Your number of cells increases, and that's what makes you bigger. When you are an adult, you have approximately 120 trillion cells. When you're a baby, you have less than that. I don't know the number. Repair means that when you get injured someplace in your body, it has to be fixed. So you get a cut on your finger, you've got a scar there from some cut that you had. The mitosis in the skin on your finger, or the mitosis in the bones in your body when they break, the mitosis causes those cells and those parts of your body to heal itself, to repair the parts and seal up any holes in your body to make sure that you stay together, essentially. All right, we're going to start our discussion of mitosis by going back to the beginning, where you had the sperm cell from your dad and the egg cell from your mom. The egg cell is big. The number of chromosomes inside of a sperm and an egg is 23 chromosomes, so that when they come together, the new organism, being the new baby, the zygote, essentially it would be you if this was the story of you, so that you would have 46 when these two come together. So they have to each have 23 chromosomes. Now, to draw and model 23 chromosomes in this process is pretty complicated, so we're going to simplify it a lot by, instead of having 23, we're just going to have 3 in each one. Sperm here, there's going to be these 3 chromosomes, and in the egg there's going to be these 3 chromosomes. Now, when these come together, it makes a new organism. Essentially, the sperm is going to go inside of here, so we're just going to move them inside of here. And this 3, the egg and the sperm fuse, and the sperm delivers its DNA to the egg, and now we have a complete set of chromosomes. If this was a model that was more accurate to the number of chromosomes being shown, there would be 46 chromosomes here. But for simplicity, we're just going to keep it down to 6 for right now. And you can see that half of those chromosomes came from dad, half of the chromosomes came from mom. We're going to use the color pink for the chromosomes that came from mom and the color blue for the chromosomes that came from dad. Okay, so our story of mitosis begins with the first stage of the cell cycle, which is called interphase. And this picture, as it's drawn right now, represents interphase. This phase is what this cell is going to spend most of its time in. There's a very short period of time that's spent in mitosis, but most of the time is spent in this phase of interphase. This is when the cell is doing its job, doing whatever its function is. You notice in the middle here is the nucleus, and I've drawn the DNA in a important phase that this DNA spends most of its time in, which is in this thinned out, uncondensed form called chromatin. So chromatin is this state, or condition, whatever you want to call it, of the DNA when it's not condensed. In this stage, it is very difficult to imagine that we're going to organize it and move it and keep it all together without any breaks or mistakes. So before the cell goes into mitosis, it has to get all of this organized in a really complete set and make sure it's all working properly. So that's what we're going to do next. Now, the DNA is going to be copied in this state right here when it's still chromatin. It's going to be copied and duplicated in what's called the S phase. So you can't see it happening here necessarily, but it's copying itself and making duplicates of itself. Now, after it duplicates itself, it has to condense. The chromatin is going to form into what we call chromosomes, and that's what we saw in the egg and the sperm. And it forms into these chromosomes. We saw these came from mom and dad. All right, so these are chromosomes. And in this way that I'm modeling it right now, they are not duplicated. Okay? This is just a simple way of modeling it. I mentioned just that before that in interphase, the DNA is duplicated before it starts mitosis. So in reality, these chromosomes would be copied, but I want to show them this way first, and then we're going to show the next way. Now, in many drawings and explanations of this, you'll see the chromosomes drawn like a little X, like this. And sometimes they'll put a dot in the middle. That's the centromere. Now, what we're showing with this type of model this way is just one side of that with the centromere here. This middle part of the chromosome right here is the centromere. So this is one chromosome. This is one chromosome. This is one chromosome, one chromosome, one, and one, and one. Each of them came from your parents. Remember, your parents each gave you 23, so there would be 46 in here if we were to more accurately model this. I can't model easily 46 chromosomes and move them all around in here. So in drawings that you'll often see, the chromosomes are going to be drawn just as a line like this, and then to show that they're duplicated or copied, it's going to be drawn like this. And we can do that in our drawing by starting off by saying that at this phase now, when the chromosomes are visible, we are going to show that their mitosis has begun. So mitosis begins in prophase. And we know prophase is, mitosis in prophase is beginning because the chromosomes become visible. Now, I mentioned that in often drawing models of this, you show it as a little X like this. We're going to show that by simply having them next to each other. So this, having two of them next to each other, represents this chromosome being duplicated. So this is a model of that drawing right there. And we're going to do that for each of these, because each chromosome has to be copied in order to go through mitosis. In order for the cell to complete mitosis, it has to make copies of all of its DNA. There we go. So now we've got duplicated DNA in all of the chromosomes, and we can say that mitosis has begun. All right, now we're going to go through these stages of mitosis. And a good way to think of mitosis, the way that I like to think of it, is from my one of my favorite biology videos on YouTube is from Amoeba Sisters, and they teach this little acronym, PMAT. This is for prophase, this is metaphase, anaphase, telophase. All right, so prophase starts with the chromosomes being visible and the nucleus deteriorating, falling apart. I'm going to show that the nucleus is falling apart and disappearing by taking the word off and showing that there's just a bit of a dotted line there now because the nucleus has fallen apart. It's important that it falls apart so that these chromosomes can all be moved around to where they need to go. All right, now that's prophase. The next stage for PMAT would be metaphase, and in that stage, the M I like to think of as meaning that they're going to be lined up in the middle. So we're going to line these up in the middle, and this is metaphase. So we already did prophase, now right now we're talking about metaphase. Alright, so if we line these up in the middle, we can put these like this. My cell is pretty small for this. I'm trying to fit these all in here in my space that I've got. If you've got a big table to work with, then that would be perfect for this. Some of my chromosomes have to be off the camera a little bit here. I'm going to cram them together. While metaphase is forming, and to get this point, there are centrioles over on the sides here. See them often drawn like little squares at a little bit of an angle to each other, one on each side. Before metaphase starts, during prophase, these centrioles are going to duplicate themselves, so there was one little pair of them, and then during prophase they copy and they move to opposite sides, and they start to grow these little arms. These are called spindle fibers, and they're going out in all directions, reaching out and reaching for these chromosomes. They're going to reach out and they're going to connect to the centromeres on these chromosomes. So remember, these chromosomes are represented as a X when we draw them. So the spindle fibers reach out and grab onto the centromere there, and the same thing would happen on this side. They would reach out and connect to this side here. So for our model with these paper models, we're going to show that they're reaching out and grabbing on to each of these little paper models here at their centromeres. This one's going to go underneath here and connect right there. This guy's going to come over here. There we go. There. It's getting messy. All right. So these are lined up in the middle, nicely organized in the middle. These are called spindle fibers. Again, these are the centrioles. Centromere. That's an O. That's the point there on the model where the chromosomes are connected to each other. This one's actually upside down right now. I gotta turn this around. There we go. So that those are they match. All right. So now that's metaphase. The next stage of PMAT is the A, which stands for anaphase. And when we're talking about anaphase, think of away. So now we're going to clean this all up, and we're going to start to pull these chromosomes apart from each other. Get rid of metaphase, because now we're going to be in anaphase. So we're in PMAT. For PMAT, we're back in the anaphase. So I'll write anaphase right here. So for anaphase, what happens is that these guys were in the middle together. Not that one, these guys here. I messed them all up. All right, so each of these was paired up with itself. This was originally this was the X, remember? And then we're going to pull them apart this way, and this one's going to go this way. So this spindle fiber reaches out and grabs onto this part of the chromosome and pulls it back this way, like that. And then another spindle fiber has reached out at the same time and starts to pull this one back the other way. So to draw that, represent that, we can put all of our chromosomes on here now. Put this one back here like this. This one can go right here like that. All right, now, so as you can see in anaphase here, these chromosomes are all being pulled apart. They had been in the middle. The spindle fibers have reached out, grabbed onto them, and they're going to pull them to the opposite sides. This is anaphase. Think of away. All right, moving on to the next stage is telophase, and at that point, these chromosomes are going to have migrated completely to the opposite sides, and the nucleus is going to start to reform around them. So let's clean this up and show what telophase is going to look like. Okay, now at this stage we're going to be at telophase. And in telophase, the chromosomes have migrated completely to opposite sides, and the nucleus starts to reform. This is when the nucleus comes back. I'm showing it as a dotted line to represent the idea that it's slowly coming back. Eventually it will be there completely. We'll have a complete nucleus in next stages as this finishes off. Notice that we finished with the same number of chromosomes that we started with. You've got the 3 from mom and the 3 from dad that you originally had before. It looks just like it did before. We had copied them before we did this, so now we still have the same number of chromosomes, the same chromosomes. Everything is identical in these new nuclei that it started off with, in just one nucleus that was in the middle before. All right, the next stage is what's called cytokinesis, and cytokinesis is not technically part of mitosis. It's simply the last step after mitosis. So PMET is the stages, the 4 stages of mitosis, and then after mitosis we have cytokinesis. So let's show cytokinesis next. So with cytokinesis we're going to form our nuclei completely here on both sides. As this is happening, the cell starts to pinch itself off. This is what would happen in an animal cell. In a plant cell it's slightly different. They form a new cell wall around the cell. But in an animal cell it starts to pinch itself off in the middle here. And it just kind of keeps pinching and pinching and pinching. And we can draw it closer now. In the next stage it would be pinched off even more, getting closer to completely pinching off. Now while this is happening though, we also have to think about what's going on in the nuclei. These nuclei are uncondensing. They're going back into their chromatin phase. So we're going to represent the chromatin coming back now to show that it's going back into interphase. So we've got our DNA from dad and our DNA from mom all going back into that stage we call chromatin. And then this side, the same thing. We got dad's DNA in here, and DNA you got from mom. Really, it's all your DNA. It's not your mom's DNA or your dad's DNA. It's your DNA or the individual's DNA. But that's just to keep track of the idea that there's 2 sets of DNA in there from each of the parents. All right, at this stage, the cell completely pinches off. This is what's called cytokinesis. Cytokinesis meaning cell, and kinesis is splitting, so splitting the cell into 2. And at that point we can say that mitosis is done. The cell cycle has completed itself. It's gone back into interphase. So this cell is an interphase, this cell is an interphase. If this cell is going to, or when the cell goes through mitosis, it's going to split into 2 more, and then it's going to be exact copies. So this cell here and this cell here are exact copies of each other, and they are copies of the cell that they came from. That's an important concept with mitosis, is that mitosis is going to make identical cells. They're not going to be different from the parent cell. These are called daughter cells. So the daughter cells are the same as the parent cells, and they're the same as each other. Okay, hopefully that helps.